It's my honor to introduce our keynote speaker, Colin Hill, who I've had the pleasure of working with over the last couple of years. We're collaborating with his company, GNS Healthcare, and the neuroscience group is working with him. Colin is a leading voice in AI and precision medicine and brings impressive leadership experience in developing and commercializing AI solutions in discovery and biopharmaceutical drug discovery and development and in managed care industries. He co-founded a leading AI biosimulation company, GNS Healthcare, in 2000, and has since served as CEO. In 2020, the company launched the world's first virtual patients beginning in multiple myeloma, and has since launched virtual patients in prostate cancer and Alzheimer's disease, which we work with Colin on. He's a frequent speaker at national and international scientific and industry conferences and has appeared in numerous publications and television programs, including The Wall Street Journal, CNBC Morning Call and Squawk Box, Nature, Boston Globe, Politico, Forbes, Wired, and The Economist, and now has Merck Technology Symposium to add to that list. He graduated from Virginia Tech with a degree in physics and earned a master's degree in physics from both McGill and Cornell University. He's a fantastic collaborator, really a very interesting guy, and has become someone I interact with on a friendly basis as well. His fun fact, amongst many, actually, I could give many that I've learned about Colin, but one that I learned just recently was that he went swimming with whales in Dominica. And he showed me a picture of this experience, and it's pretty amazing, the size of the whale. My gosh. If that's not bravery, I don't know what is. Speaking of bravery, he has invested in this new area of research, pairing artificial intelligence with biology, and I think his presentation will be provocative. He meant it to be provocative, and hopefully give us a flavor of what can be done with artificial intelligence and how we're going to reimagine discovering new target identification and validation. So with that, Colin.
Good morning. So I'd like to thank Jason for that very kind introduction, and I do plan to make it back to Dominica to do more swimming with whales sooner than later. I'd also like to thank you, Jason, and Amy and Marissa and Victor for inviting me to participate in this year's event and for all the work you did in organizing it. I'm really excited to be here today, given the recent developments in the industry. We're living through a very interesting period with genomic profiling, single-cell data generation, new model systems, artificial intelligence, and now these trends are starting to converge. And they're converging in a way that's making it possible to do the impossible. Specifically, I'm thrilled to be collaborating with Jason and the teams at Merck to actually start to apply and bring some of these developments to fruition. Today, I'm going to discuss a topic that's becoming a more critical part of the drug discovery industry. And that topic is, can virtual patients unlock the biological complexity of human disease and drive the next wave of breakthrough drugs? The answer is yes. And we will have new drug candidates from virtual patients in clinical trials within the next 4 years. It's a very bold assertion. But with the data and the evidence I'm going to share with you today, I think you're going to come to see that the time really is now. So what are virtual patients? Virtual patients are computational representations of a disease that captures a sufficient number of the genetic and molecular interactions that drive clinical and physiological outcomes. As you can see in the figure on the right, the genetic variables here are driving the molecular variables that in turn drive clinical outcomes. And so the idea here is that these virtual patients are really capturing the key biology driving a disease, essentially creating a digital twin. Sounds crazy. What can you do with these virtual patients? With these virtual patients, you can essentially now perform computational experiments, knocking out genes and proteins one by one to discover novel drug targets that can then eventually turn into new breakthrough therapeutics. You can also simulate disease progression and drug treatment at the individual patient level, essentially allowing you to simulate clinical trials before running the trial in order to now discover which patients are going to respond or not respond ahead of the trial. Imagine that. These patient groups who respond being defined by specific biomarkers contained in the virtual patients. So is this too good to be true? I don't think so. With the growing quantities of data, the growing amounts of computing power, and now the reduction to practice in specific diseases, virtual patients are becoming accurate enough to be used as a critical part of the drug discovery and clinical development process. The impact will not be incremental. The impact will be dramatic, and the impact will be transformative. So why has this been so difficult to create virtual patients? In other industries, such as aerospace or the semiconductor industry, we're used to predicting the behavior of complex systems all the time. What makes human disease biology so special? What you see here on the screen is a blueprint of an early computer chip. What to take away from this is that with all the connecting transistors and capacitors that interact with each other through the flow of electricity, these computer chips work. And today, it's pretty routine to have computer chips have tens of billions of features on them. And yet, we're able to make computer models of computer chips, right? Essentially digital twins, equivalent to our virtual patients, that work. It works to predict the behavior of these systems very reliably. It's driven a crazy amount of value in these industries to the point where we take it for granted. So why not in human biology? Why not in human disease? Here is a blueprint of the genetic circuitry of cell replication in cancer. What you're looking at are interacting genes and proteins that control the replication of cells. So these systems have thousands, tens of thousands of components. Remember, we have 30,000 genes. So you could say, well, this is many fewer components than computer chips. So why can't we easily predict what's driving new cancers or Alzheimer's or other diseases? Why do we struggle so much to figure out what things to hit and in which patients they're going to work in? The answer, despite all of this knowledge we do have after 70 years of molecular biology, is that after those 70 years, we are still missing 95% of the circuitry. Let me repeat that. 95% of the circuitry of human disease is unknown. So what you're looking at is just the tip of the iceberg. And so our jobs collectively, right, is to discover new drugs, to put those through clinical development, despite the fact that we're missing 95% of the circuitry. Makes our jobs a lot more difficult than the guys at Intel. So what to do about it? Why did prior biosimulation efforts fail? So GNS is not the first company to try to create virtual patients and simulate human disease for the sake of discovering new drug targets and simulating clinical trials. In the late 90s and the early 2000s, companies such as Entelos, Archimedes Model out of Kaiser Permanente, and Physiome Sciences attempted to make virtual patients and digital twins. I'm guessing Merck probably even collaborated with some of those companies at the time. So why did those efforts fail? It goes back to what I just said. Those approaches were only based on the existing 5% of known knowns contained in the literature. And at the time, there was scant other data to really supplement that with. So these groups had the right idea, but we didn't have the data, and we didn't have the opportunity to try to fill in this missing 95%. So why now? What makes us think that the world is different now and we can actually create accurate virtual patients to drive a whole new generation of drug discovery and clinical development? Number one, we've been enjoying the exponential growth of multi-omic plus clinical data directly from patients. So not just mice, not just in vitro systems, but directly from patients where the disease exists in. On the left, you see a graph showing the cost of the human genome dropping precipitously as the speed has gone up. So in 2001, with the sequencing of the first genome, that took 11 years at a cost of $3 billion. Today, that same genome is sequenced in less than a day and less than $1,000. The same is true for proteomic and gene expression technologies, or at least it's getting there. And that's giving rise to now large-scale multi-omic and clinical data collection efforts such as the UK Biobank, the Asia Biobank, disease-specific efforts such as ADNI and Alzheimer's and AMP-AD. So we now have the data on a very different scale than before. And over the next 2 or 3 years, the amount of data coming from these efforts is going to increase by orders of magnitude. That's a fact. Number two, we've been able to enjoy quietly decades of Moore's Law that have given us near infinite computing power. Moore's Law predicts the doubling of computing power every 2 years. So that has consistently been going on for decades, giving us a huge amount of computing power that allows us to crunch data on a scale that just was not possible 20 years ago. We would have dreamed of this kind of computing power. Thirdly, there's been a shift in AI, and specifically the emergence of causal learning. Deep learning is about finding correlations and patterns in high-dimensional data sets. Powerful for a lot of things, and in medicine, powerful for imaging. But that's not the same as cause-effect relationships being extracted directly from data. Right? So causal learning is a much more powerful approach to now getting at the mechanisms of the system that created the data. So you can say, why is this so important? Isn't all AI just AI? And the answer is no. The concept of extracting causal relationships from observational data was considered heresy for many decades. Until pioneers such as Judea Pearl came along and showed that in fact, this is the case, that one can extract probabilistically cause-effect relationships from data. And for his pioneering work, he was awarded the 2011 Turing Prize, the equivalent of the Nobel Prize in computer science. To reach the higher fruit, AI needs a ladder, which we call the ladder of causation. Last year's Nobel Prize in economics went for the science of causality in natural experiments. And even some of the pioneers in deep learning are now coming around to see causal learning as the next true frontier. This is where AI comes back to meet science, mechanistic science, biological science. So how do we think about the difference between correlation and causation? There's a little story I like to tell that I think illustrates the point. There's this very smart robot named Larry. Larry is infinitely smart. It can learn very quickly, but Larry doesn't know anything to begin with. Larry's dropped down in the cornfields of Iowa, and his job is to learn about the world around him. The first day, he sees the sunrise at the same time as a rooster starts to crow, cock-a-doodle-doo, at exactly the same time. Day 2, the same thing. The rooster crows, the sun rises. Day 3, day 4, the same thing. So Larry is clearly seeing this correlation. He's seeing this pattern between the sun rising and the rooster crowing. But what Larry doesn't know is what's causing what. Is the rooster causing the sun to rise, or is the sun causing the rooster to crow? Sounds like a silly thing to say because, of course, all of us know the direction of things. But remember, Larry doesn't know anything, and he's learning, just like the way babies and toddlers learn. So the question is, how does Larry figure out what's causing what? Don't be shy if you have the answer. So if you guessed that Larry blindfolds the rooster and puts the rooster in the basement, you're right. That's exactly what Larry does. And then he waits to observe, does the sun rise the next day or not? Lo and behold, the sun rises the next day, causing Larry to conclude that the sun must have been causing the rooster to crow. So Larry discovered causality from observational data. Now, this observational data, as a result of these natural experiments or interventions, happen all around us. And the question is, can we learn those causal relationships straight from that data? Unlike correlation, which is asking, is A related to B, causal inference is testing in parallel a vast number of hypotheses of the form, does A cause B or does B cause A? It sounds simple, but it's very profound. When you start talking about thousands of variables, it becomes transformative. So correlation is about answering the question of what happens when I see. Causation is about answering the question of what happens when I do. So to sum up, the convergence of multi-omic data straight from patients, near infinite computing power, and the emergence of causal AI at scale is allowing for the creation of cohorts of virtual patients in a way that was not possible 20 years ago or 10 years ago, or even really 5 years ago. So how to create these virtual patients? How does my team at GNS actually do this? It sounds good, but how does it work? So the initial steps in creating virtual patients are as follows. It starts with the data. And on the left, you see here data from the Multiple Myeloma Research Foundation, one of our collaborators, and specifically the COMPASS study, which is the largest multi-omic and clinical data collected ever in the world in multiple myeloma. It's from 1,100 patients, 90 different sites. It includes whole genome sequencing, RNA-seq, miRNA, immunoglobulin profiling, cytogenetics, and clinical parameters. That data is now placed into a data frame showing the values of the genes and the other molecules and the clinical outcomes for each patient, one by one by one. From that data frame, the building blocks are computed. Think of this as multivariate regression on steroids, where all of the two-way and three-way and higher order interactions are computed. And you can see on the right, A and alpha together driving the value of C. You see that little symbol inside the horizontal triangle? That's indicating the mathematical form that's being tested to determine if these interact. And so this kind of process produces now trillions of building blocks with corresponding p-values, or the probability of that relationship existing given the data. So now you have this big bucket of essentially jigsaw puzzle pieces created from the data. We then use those pieces to now drive the optimization process, where all these pieces are stitched together into a global model of the system to create a causal hypothesis. Each causal hypothesis is then evaluated against the original patient data for its ability to reproduce that data. Each evaluation gives it a score and causes that causal hypothesis to change either in big ways or in small ways. Parts are ripped out, other parts are added, and it gets tried again and again and again. Now, this process is happening with thousands of different causal hypotheses on thousands of processors simultaneously in order to now extract, after evaluating many trillions and trillions of network hypotheses, it's now attempting to extract an ensemble of 1,000 best performing networks. These are the networks, the virtual patients, that are the best representations of the system that created the data. And that's what you're seeing on the right. And this bigger picture is showing how this process is filling in that missing 95% of the circuitry and simultaneously reconstructing the known biology. The known biology is represented, as you can see, in black, and the unknowns that were being discovered are represented in red. So this process is happening in real time as our systems are now trying to stitch together each of the causal hypotheses and determine what is the most representative of the system that created the data. So this is very different than pattern finding. This is not deep learning. This is an attempt to extract the biological mechanisms straight from data. Crazy concept. People have spent their PhDs trying to discover any particular link. And now I'm telling you that we are starting to discover a whole bunch of links computationally straight from data. So when these virtual patients are created and there's sufficient data to make them converge, we can now use them to discover novel drug targets. How do we do that? We take the ensemble of models. Keep in mind, there's not a single best fit model. We take the ensemble of models and we simulate gene knockdowns across the board. And what you're looking at on the left side of the screen is one particular molecule that when its knockdown is simulated, it's shifting the clinical outcome from the red histogram to the blue. It's reducing the size of the tumor in this case, represented by cell count. And why is that? You can see that that molecule is on the causal pathway to the clinical outcomes, right? On the other hand, this other molecule on the right-hand side of the screen, while it might be strongly correlated with the clinical outcome, and it might have a common genetic driver upstream, it is not on the causal pathway to the clinical outcome. And when that's knocked down, you can see on the right that it does not move the clinical outcome. So that would make for not a very good drug target, even though it might look really interesting. So computationally, we are now performing essentially siRNA knockdown experiments and doing it at scale, and this is becoming more and more accurate. We're also using these virtual patients to simulate drug treatment at the individual patient level, allowing us to start simulating clinical trials to discover responding versus non-responding patient populations up front. And here you're seeing the shift of a clinical outcome from the orange phenotype to the green, showing that this drug is indeed having a major effect on patients with a certain genetic background. Okay, so now I've spoken conceptually about how this all works. And I'm sure half of you are really skeptical right now still, and you want to see a real example. You want to see the meat. That's exactly what we're going to do. And in particular, how this was applied in the area of cardiovascular disease to discover novel drug targets driving atherosclerotic disease. Atherosclerotic disease remains the #1 cause of death in the United States. Lp(a) has emerged as a causal factor for the development of this disease. A patient's Lp(a) status is largely genetically determined, and it is the strongest genetic lipoprotein abnormality and the strongest genetic predictor of myocardial infarction or heart attacks. However, what drives the elimination of Lp(a) from the bloodstream is still unknown, and there's conflicting experimental evidence. The data that fueled the creation of the virtual cardiovascular disease patient came from our partner Global Genomics Group, or G3, and the global study. This is the single most comprehensive multi-omic data set that's anchored in genetics for both cardiovascular and cardiometabolic diseases. It was collected from 48 clinical sites, 7,700 patients, trillions of data points, and included whole genome sequencing, DNA methylation, RNA-seq, proteomics, and so on. We had an equal amount of controls and cases in this study. And as a constraint in the reverse engineering process, the clinical parameters in the DNA sequence variants were held as drivers of the molecular layers of the DNA methylation, miRNA, mRNA, and so on, driving the clinical endpoints. And here's what emerged. You see at the top in purple are the genes coding for Lp(a), the LDL receptor, PCSK9, and some newly discovered genes that drive the multi-omic network, the causal network in green and blue and orange, that in turn drive the clinical endpoint. What you see in this process was the reconstruction of 60 years of cardiovascular disease biology done in the course of 3 months. Really amazing in terms of the biology contained around PCSK9, LDL, and statins, and so on. What's even more interesting was the discovery of some novel biology driving the regulation of Lp(a), which we're going to zoom in on here. So what you're seeing, the arrow above is showing and is pointing to what was discovered in the virtual cardiovascular patient, which is the fact that smaller Lp(a) particles are being cleared via the LDL receptor. However, the larger Lp(a) particles are being cleared by a different receptor, a newly discovered receptor from this process. So this is still unknown, which is why that particular receptor is being covered up by the gray box. So this novel discovery of this target, as you can see, was also genetically confirmed, where you're seeing on the right, now this is a genetic perturbation of that particular receptor, and it is in fact impacting the Lp(a) levels. Working with a major pharma company and G3, we went about trying to validate this new hypothesis. This hypothesis, just to be clear, is that the small Lp(a) particles are being cleared by the LDL receptor that you see on the top, whereas the larger Lp(a) particles are being cleared by this novel receptor that's expressed on monocytes, adipocytes, and endothelial cells. The experiment that was then conducted was doing siRNA knockdowns of this substrate So this was now knocking out this novel receptor and observing its impact on the natural ligand for that receptor, as you can see, is decreased by 25%. That's substrate 1. Substrate 2 is Lp(a). And as you can see, the siRNA knockdown is resulting in a 20% reduction in the uptake of Lp(a), confirming the discovery of the impact of this novel receptor on Lp(a) levels. While there's more to be confirmed with this result, this is potentially an important breakthrough in cardiovascular disease medicine. And keep in mind, it was discovered in a hypothesis-free fashion, straight from these virtual patients and this super large data set. Now let's talk about Alzheimer's. And this work is the result of a collaboration between Merck and GNS, which is still ongoing. And it's been very exciting. And I think we're really just getting going with it, meaning there's a lot more that can be done. Just as a reminder to everybody, Alzheimer's disease is the 7th leading cause of death in the United States and the most common cause of dementia in older adults. More than 200 programs have failed or been abandoned in the last decade. It's been one of the most difficult areas in all of biomedicine. Merck and GNS have been collaborating to leverage and integrate multidimensional data sets directly from human patients and apply GNS causal AI to create virtual Alzheimer's patients that capture the complex interplay of genetic and molecular factors driving the physiology and clinical outcomes. And we're doing this for the sake of discovering truly novel drug targets that can one day turn into novel therapeutics that are going to be effective against this horrible disease. That's the reason. The data sources came from both blood and brain. So the blood data source was ADNI, which is the Alzheimer's Disease Neuroimaging Initiative. It's a longitudinal multi-center study designed to discover novel biomarkers for the disease. It's blood-based, comes from, this particular data set comes from 300 patients, and as you can see, it had huge amounts of multi-omic profiling contained in it. We did not want to depend on just one data source, so we simultaneously used post-mortem brain tissue from the AMP-AD effort, and that was 800 patients and a large amount of multi-omic data. What you're now looking at is the reverse engineered network in Alzheimer's disease, where you see the gene expression variables in blue, representing potential drug targets, demographics in green, driving the biomarkers and the clinical outcomes. In this process of reverse engineering the virtual Alzheimer's disease patient, we, of course, reconstructed and rediscovered key drivers such as the tau proteins, ApoE4, and others. The reconstruction of that biology is really important because it really confirms that we're on the right track. And then simultaneously, of course, we've discovered some novel drivers of these outcomes. This is now the same network, but now in the postmortem brain tissue, where you see the gene expression variables and demographics driving the biomarkers and clinical outcomes. There was a large number of potential drug targets found at the gene expression level, both for the blood-based models on the left and the direct brain-based models on the right. One particular gene, one particular target is target X, which we discovered as a driver of tau-related outcomes in both the blood and brain-based models. And what's really interesting about this novel target is that it's driving the tau biology in very particular ways in both systems. And in particular on the right, you can see its impact on A beta and other now clinical outcomes downstream of that. Experiments that were being conducted at Merck in the area of ER stress. It's a particular fundamental part of biology that underpins a lot of neurodegenerative disorders. The experiments that were being conducted by Merck overlapped with 53 of the predicted genes from GNS, and 9 of those actually validated. 9 of those had now wet lab confirmation. So from experiments that were being done in parallel within Merck, 9 of these completely novel predictions from the GNS virtual patients were now confirmed, now had hits. A lot more work to do, a lot more validation to do, but this is a very exciting starting result. One of those particular targets was found now to drive several other targets of ER stress in the same virtual patient networks, driving 2 clinical outcomes. And so now we're able to put these novel targets in the context of the biology and of the biological network contained in the virtual patient. This has been a very exciting collaboration so far, and we have super high hopes for where this goes in the very near future, both in Alzheimer's and beyond. So I've now showed you 2 examples of where these virtual patients are used to discover novel drug targets. Can we use these to discover and to simulate clinical trials? And the answer is yes. And we've done this in the area of prostate cancer. So prostate cancer is the second most common cancer in men. More than 400 trials have been conducted in the last decade, with very few getting approved. So the challenge is to better select patients in these trials. We use data coming from Tempus, our partner, TCGA, and Sloan Kettering. And this is very deep multi-omic profiling from both germline and tumor that led to the construction of a virtual patient. You're seeing the full virtual patient on the left, and a subnetwork on the right centered around the androgen receptor, which is now driving some key areas and key pathways of biology, P10, P53, E2F1. And it also shows the impact of various drugs like enzalutamide, abiraterone, on the overall survival endpoint. We then simulated this virtual patient first to see, can we reconstruct recent clinical trials? So what you see on the right is the result of simulating abiraterone versus standard of care. And the blue histogram is showing the hazard ratio across overall survival. And what you can see is that it's centered more or less at the same point as the orange horizontal line, which is the actual result of the trial in the error bars. So in both the case of abiraterone and enzalutamide, it's reproducing directionally and in magnitude the result of these 2 trials. We then use these same virtual patients to now discover 2 germline mutations that are driving response and non-response to enzalutamide. And this was presented at ASCO, the American Society of Clinical Oncology, just a few months ago, which showed a novel discovery of a particular subgroup of men who do not respond to enzalutamide. And so this is showing that we can start to use these virtual patients in clinical development. Now, to wrap up my talk, I want to ask even a more provocative question, which is, can we eventually use these virtual patients to impact patient care? So with enough data and more data feeding these virtual patients, the answer is yes. And it must eventually have this impact. All of us, we do what we do for a number of reasons. But one of those reasons is to impact patients. Sometimes that's even family members and our loved ones. I've had a couple of recent incidents where these challenges really came home to roost. I used to write a blog for Forbes about 12 years ago on Healthcare 2020, when 2020 seemed like a far way away. And during that time, my father was diagnosed with advanced prostate cancer. He had, at best, a 50-50 chance of survival and of responding to standard of care. And so in this journal, I chronicled our challenge, our journey. Can big data save my dad from cancer? And so we used the most advanced DNA sequencing of tumor biopsies that were available at the time from Foundation Medicine and the advanced analytics, essentially a precursor to a virtual patient, to determine if my father did not respond to standard of care, was there an alternative that could work for him? We did not find anything, but my father did get lucky, and he responded to Lupron. and he's still with us today. This is my cousin, T.D. Hill, who 2 years later was diagnosed with advanced cervical cancer. We once again tried the most advanced techniques possible to find a miracle cure for her, something that was going to work where standard of care wasn't. And this time we weren't so lucky. It did not work, and my cousin passed away 2 months after she was diagnosed, despite doing everything possible. I believe, and I sincerely desire, that we can change results for people like my cousin and other patients around the world with advances in technology and data like what we've talked about here today. I do want virtual patients to be able to help people like my cousin in the near future, and I believe it will. I sincerely believe it will. You've been a very patient audience in talking with me about really the transformation of our industry from an industry that's largely been based on chemistry and biology, one that's now becoming fused with mathematics and computing in a very different way. The world is changing. We now have the power to start to unlock the underlying complexity of biological networks underpinning human disease. This is not going to be an incremental change. We really do have the power to unravel the underlying biology driving disease and start to discover and develop new drugs in a way that's much more predictable and powerful and better matched to patients than ever before. We have to unravel the missing 95% of the circuitry in order to do this. And fortunately, we now have the tools, we now possess the tools to do this. And it does require deep collaboration across chemistry, biology, mathematics, and computing. And I assure you that this will change everything. Thank you very much. I think we have a minute or a few minutes for some questions.
We do. We'd like to invite folks in the audience to approach one of the 3 microphones if you'd like to provide a question to Colin. Thanks.
Thanks. Very nice talk. Thank you. I've got 2 questions. The first one is, in your pathway sequence from gene to outcomes, I don't know if it was just coincidence in the drawing or if there's something significant there. None of the pathways, the simulated pathways, look like they went completely from gene to outcomes. They always seem to incorporate at least one step of a known step. Is that coincidental in the drawing, or is that a requirement for this to work?
Yeah, it's a great question. That is coincidental in the drawing. Although in reality, oftentimes, some of the known biology is going to be involved in terms of the reverse engineered circuitry that goes from target to clinical outcome. And so it certainly isn't a necessity, but that's often going to be the case. I mean, the known biology that we've discovered does represent a lot of the really important biology, because those were the first things that popped out.
Okay, thank you. The second question is, have you looked at applying this approach to off-target activity, toxic pathways, and what are the challenges there?
Yes, the answer is yes. So we have applied this to now predict safety outcomes, adverse events, and the like, especially because from this approach and based on multi-omic profiling, you don't have to depend on the known mechanism of efficacy that we think the drug's working through. We now have the ability to discover what the drug's actually doing and discover those off-target effects that are driving potentially bad outcomes, and to do that early. So great question.
Thank you.
Thank you. Nice talk. My question I had was, because a mathematical causation is not the same as a clinical causation, and because the regulatory agencies, they strongly emphasize validation of systems, I was wondering, what's going to be the long-term regulatory validation strategy? Because when I think of validation of a model, I think of some boundary condition. We test within a certain boundary condition, the model works. So what would be the boundary condition here? Is it going to be initially more limited? Or if you want to apply the model more widely, how would you validate it? Yeah. Thank you.
It's a great question. So, I mean, first off, I do believe the regulatory acceptance of these approaches has to be disease by disease. I do not think the FDA is going to give blanket approval for virtual patients writ large. That's number one. Number two, it does depend on the limits and the boundaries of where this works, does depend on the specifics of the patient population. So in any disease, whether it's multiple myeloma or prostate cancer or Alzheimer's, you're going to have different subgroups. And so I do believe the approvals are going to be within the sphere and the limits of the models, which is really driven by the data that's input into the models. And so those are really going to be the two constraints. I do strongly believe the FDA will start to adopt these approaches more and more, just like they did with PK/PD.
Thank you.
Okay.